Good morning, everyone. So my name is ALNAJJAR. I'm a PhD student at the University of Lorraine. I'm going to be presenting to you today our work on the fate of chlordecone under anaerobic digestion. Chlordecone is an insecticide that has been extensively applied in the French West Indies between the years 1972 and 1993 against the banana black weevil. It's a persistent organic pollutant that accumulates in the food chain and triggers chronic toxicities in a human. It currently contaminates the majority of the environmental compartment in the French West Indies, from soils to groundwater and surface waters. For this, maximum residual limits have been established by the authorities for every food type to protect the population. In the example of farming, which is an important economic activity in the French West Indies, the farming has been intensively affected by the contamination with chlordecone. In the example of the outdoor reared animals, chlordecone gets accumulated in the tissues and organs of the animal who are continuously exposed to chlordecone. And if the chlordecone concentration exceeds the maximum residual limit, then the animals cannot be consumed by human. Recently, a depuration period or a decontamination period has been proposed where the animal can be moved to a non-contaminated soil until the concentration of chlordecone drops below the maximum residual limit. Chlordecone gets metabolized in the liver to its alcohol form, chlordecone, both of which can be then excreted mainly through the fecal route. So this depuration or decontamination period is actually an excretion of chlordecone and chlordecone from the organisms. This is why it is important to find some strategy to treat the contaminated animal effluents in order to simply avoid the translocation of chlordecone from contaminated to non-contaminated soils. One strategy to treat the contaminated animal effluents is to degrade the contaminant. Even though persistent in the environment, several lab-scale studies have demonstrated that chlordecone could be efficiently degraded in the presence of certain microorganisms, mainly under reducing conditions. And these studies have succeeded to identify different transformation products belonging to different families of transformation products, such as the family A, the hydrochlordecone, with substitution of chlorine atoms with hydrogen atoms, families B, C, D, and E, the polychloroindenes, with an opening of the ring structure, which is partly responsible for the persistence of chlordecone in the environment, and the sulfur derivatives with family F. We wanted to test whether the anaerobic digestion could actually degrade chlordecone and therefore be considered as an effective treatment strategy to treat the contaminated organic waste. The anaerobic digestion has been, a lot of different speakers have already talked about anaerobic digestion during this symposium, so I'm just going to skip this general description of AD. To test our hypothesis, a digestate from a mesophilic reactor that was originally fed with animal effluents was spiked with a solution of 20 milligram per liter of chlordecone and incubated under mesophilic condition, meaning at 37.5 degrees, for 40 days. In parallel, another digestate from a thermophilic reactor that was originally fed with organic waste from plant origin was also spiked with a 20 milligram per liter of chlordecone and incubated under thermophilic conditions at 55 degrees. We wanted first to make sure that the introduction of this contaminant did not alter the anaerobic digestion process. So we followed basic operational parameters and we found a similar biogas production, similar pH and redox potential values between the bottles spiked with chlordecone and the bottles that were not spiked with chlordecone, the negative controls. So we could say that the anaerobic digestion process wasn't actually affected by the presence of such a high concentration of chlordecone. Now, in order to investigate the fate of chlordecone in our experiment, an analytical protocol was specifically designed and tailored for the identification of chlordecone, but also its transformation product in the complex digestate mixture. So after each incubation time, the bottles were extracted using a modified Quetcher's protocol, and analyzed using both gas and liquid chromatography. As for the result, under mesophilic condition, a slight decrease of chlordecone by around 40% was obtained after 40 incubation days. While under thermophilic conditions at 55 degrees, a more important decrease by around 80% was observed after 40 days. Now, by looking at these results, we cannot simply yet conclude that chlordecone has been degraded, because chlordecone has a strong affinity to organic matter. So this decrease could simply be the result of a strong adsorption of chlordecone onto the organic matter present in the digestate. However, we were able to detect and quantify several transformation products belonging to different families of transformation products under both conditions, mesophilic and thermophilic, such as the monohydrochlordecone, A1, and 2 dihydrochlordecanes. So these are transformation products belonging to family A. We also found B1, B3, B4 belonging to the family B of polychloroendians, and C1, C2, C3, C4 belonging to the family, transformation product family of polychloroindian carboxylic acids. This transformation product increased with the incubation time, and as you can see in the thermophilic conditions, the concentration of transformation product was greater compared to the mesophilic conditions. Now, in the mass balances of our reaction, we can clearly see that the decrease in concentration of chlordecone was coupled with the increase in its transformation product, proving that chlordecone has been degraded under both conditions with a more efficient degradation at 55 degrees. Now, we also wanted to test the fate of chlordecone, which is present also in the animal effluent under anaerobic digestion. So we followed the same protocol. We added chlordecone, but we also wanted to test the fate of the major obtained transformation product, which are the A1 and the B1, also under anaerobic digestion. We wanted to see if this transformation could be further dechlorinated or transformed. Of course, chlordecone was added as a positive control, and now the analysis of the bottles is in progress. To conclude, under our conditions, experimental conditions, chlordecone was degraded under anaerobic digestion in both mesophilic and thermophilic conditions, and the degradation was evidenced by the decrease in the concentrations of chlordecone and the increase of its transformation product. A more efficient degradations of around 85% was observed under thermophilic conditions compared to just 40% under mesophilic conditions, and this was only after 40 incubation days. What we want to do next is to try to identify the microorganism that could potentially be involved in the degradation of chlordecone. And next, try to test the applicability of our method by studying the fate of chlordecone and chlordecone under anaerobic thermophilic digestion, but this time in actual contaminated feces, not just spiking the solutions into the digestate, and a continuous mode using a continuous lab-scale reactor. Thank you for your attention.